So maybe that was incorporated into culture because people felt better, felt good. And actually that prepared them for the real starvation that came later. This preconditioning is something that we do when we do exercise all the time, right?
Right.
Because we do low-level exercise, we are prepared for the high-level exercise.
Right.
That training, this preconditioning of the body, is something that is good for your neurons, good for your astrocytes, that cleans them from stuff that is
Yeah.
not
Yeah.
wanted.
Yeah. It makes a lot of sense. Yeah. If the first time you have to sprint is to run away from, a saber-toothed tiger, you're probably not going to outrun it. But if you've been preconditioning, you can do that. And you can sort of just generalize this concept to a lot of different contexts. So, yeah, this idea that so of starvation, really, ketosis being a signal of potential starvation, you're going from glucose-based metabolism to using things like BHB more. Can you talk a little bit more about what is going on in the brain and in neural networks when these starvation indicators are coming online and you're starting to go from glucose to BHB, you're starting to go into ketosis? In terms of the signaling and how the neural networks are being reconfigured, functionally, how are they changing? How does that tie into maybe sort of the ecological needs of an animal in a starvation or pre-starvation state?
So we have a few answers on the cell level. So people have thrown ketone bodies to cultures and slices and see what happens with the currents, with the cell functions. And it is very much like lactate. So you have a hyperpolarization of neurons. So they become less active, they spend less energy. And this is through activation of a certain specific type of potassium channels, the KATP channels.
They're basically becoming less excitable and therefore sending out fewer action potentials. And because even one action potential takes quite a bit of ATP, this you can immediately interpret that as sort of an economical or energy conservation mode of being.
That's right. You go into less energy mode. I don't know whether you have done a little bit of fasting yourself, but when you do that, I remember trying the intermittent diet a few years ago just for fun, and you really feel whacked. So you feel like it's not working at the beginning, and that correlates with this depressing effect of BHB. At the same time, the BHB effect on astrocytes is to depress glucose consumption. Mm-hmm. So in other ways, the BHB pushes glucose away and becomes the main fuel for many cells. Never 100%, but to a certain extent, which would be 30, 40%. So BHB displaces glucose, so the cell will use BHB now because it's a nice fuel. And you burn the BHBs to CO2 as you did with glucose before, and the cell can use glucose for purposes which are more strategic. Say, in the case of neurons, glucose is not just used for energy. Glucose is very important for protection against reactive oxygen species. So one of the first pathways when glucose enters glycolysis is that it's directed to the side pathway, which is called the pentose pathway. And the pentose pathway produces NADPH, which is the main antioxidant of the cell.